My assignment at the World Soybean Research Conference was to sort of give an overview of genomic selection and genomics-assisted breeding, mostly genomic selection, to set the stage for 4 fantastic speakers that were to follow me, but they're not here, so you just have me. So with that in mind, this is a typical variety development breeding pipeline, right? And this happens to be our pipeline at the University of Minnesota. Where should I stand here? And if we perform this pipeline correctly and we make correct selections like we should, if we're doing everything like we should, then we're going to get genetic gain, right? We select the best parents, gradually move forward, and over time we improve really complex traits like yield. That's our job as plant breeders. So in terms of breeding methodology research, there are lots of ways that we can make this pipeline better, faster, more effective, more efficient. And 2 current popular areas of breeding methodology research, they're not the only areas of breeding methodology research, but 2 currently popular ones are genomic selection and high throughput phenotyping. And I don't want to call these things trendy because I think these are really important breeding methodologies that are here for the long term. And we have activity in both of them. And I'd like to come back someday and talk about a lot of the great research we have going on in high throughput phenotyping. So, but the goal of of applying these technologies, these methods to a plant breeding program is essentially to make that funnel that we have wider and shorter for the same amount of cost. Basically, get more material through our pipeline that costs the same, and shorten up that time both to parent selection and product development. That's the basic goal. So this is genomic prediction in a nutshell. So genomic prediction in a nutshell, we have DNA marker data. We may have thousands, tens of thousands, hundreds of thousands of SNPs or DNA markers, whatever they might be. We combine those with phenotypic data for a trait of interest, typically a complex trait like grain yield, maturity, maybe a complex disease resistance. We put those together, we call it a training population or calibration set, right? And then we put those things together and we build a statistical model to enable predictions. We apply that statistical model to individuals that we call selection candidates that have been genotyped but not phenotyped, make the predictions, and then we use those predictions for selection. Maybe it's using predictions for selection for rapid cycling, choosing parents much, much earlier than we normally would. Maybe it's to augment single replication or very shallow phenotypic data to get a better idea of the true genetic value that we want to estimate. So the difference between this and using what we typically call just plain old marker-assisted selection or traditional marker-assisted selection is that in this case, we're not doing any QTL mapping. We're not first going in and finding, okay, which parts of which chromosomes affect the traits and only use those. We use all the marker data simultaneously without any discrimination of which ones are the important ones and which ones are not. And that's because, as we know, and we've probably known for a long time, we had to rediscover that complex traits are controlled by many, many small effector genes. Right? It's really hard to find just a handful of QTL that control most of the variation. We heard today that even disease resistance to STS is a highly complex trait. So you can imagine how complex something like yield is. There's not a single, or there are not a handful of QTL that we can really manipulate. Rather, that we model all the marker information simultaneously. And again, just looking at the literature here, genomic prediction compared to a marker-assisted selection approach typically works a lot better, all right? And it always works at least as well as marker-assisted selection. So if you compare marker-assisted selection here to prediction accuracy, this is prediction accuracy, or in other words, the correlation between the prediction and the actual observed phenotype. It always works better than a marker-assisted selection approach in these, I think, 36 combinations of populations and traits that Rex's group looked at several years ago, and that's a simulation study of mine a few years ago, and this has been backed up in several other studies as well. So it's a good approach for dealing with complex traits. Now, surrounding this process, there are lots and lots of questions. How do we exactly optimize this process? So we can look at things like, what marker platform do we use? How do we minimize marker costs? And just the pure logistics of collecting that many markers on that many samples. Now that I run a breeding program and I'm faced with 10,000 progeny rows, for example, just extracting DNA and collecting marker data on 10,000 progeny rows is not a trivial process. There's all types of questions around that. How do we optimize that? How do we actually do that in companies have this figured out, but I think a lot of public programs still don't have this figured out very well yet. There are questions surrounding just training population design. So which individuals do we put into our calibration set to make our calibration set as informative and predictive as we possibly can? And on top of that, which model do we use? And how do we model genotype by environment interaction effects? Those are just a few questions that pop up. And I'm not going to cover all of these because I was only given about 20 minutes or so. So I'm just going to cover a couple of these things. So I'll cover model here real quickly. So in a typical genomic prediction type of scenario here, we typically have many, many more markers than we actually do observations, which creates this problem called the large p or small n problem, or the curse of dimensionality. Right? We don't have, even if we wanted to fit a traditional linear model, we don't have enough degrees of freedom to estimate all those effects. And even if we did have enough degrees of freedom, we have all kinds of problems with multicollinearity, which is going to create an overfit model. Right? So folks in this field applying genomic data to predictions have both mostly adapted, but also developed a lot of specialized models for this problem. And there are lots and lots of models out there that I'll describe a little bit of. You know, genomic G-BLUP and R-BLUP are very, very popular models, but there are also lots of Bayesian models, lots of machine learning types of models. Basically, these models differ by what types of assumptions do they assume about the underlying marker effects, as well as the interactions that they may try to capture. So these have basically been developed to get around that problem. So, but the good news is, I guess it's good news, depends on which way you look at it, is that a lot of these models don't perform any better than what I'll call the kind of the standard model, the G-BLUP model, or its equivalent, the ridge regression best linear unbiased prediction model, the G-BLUP and the R-BLUP model. These are what I think are the simplest models. And all these other models, at least in the literature, in a plant breeding situation, don't really perform that much better for a typical complex trait. This is, I went through the literature quickly the other day and pulled out all the soybean genomic selection papers. There aren't that many. There are only about 6 or 8 or something like that. All the models have actually compared G-BLUP to an alternative, more complex model. Okay? And I plotted out the comparison between the prediction accuracy of G-BLUP versus the alternative model. Right? And you can see that they pretty much line up on that one-to-one line. And what I have highlighted right here, this is what I call the who cares zone. Right? This is not a statistically validated concept, but it's basically plus or minus about 0.02 correlation points or something like that. So it's really within that, not even statistically significant, and even if it were statistically significant, who really cares? So they pretty much all line up right there. So that's good news, depending if you're a model researcher or a plant breeder like myself. So a little bit about G-BLUP. What exactly is G-BLUP? Like I said, G-BLUP is mathematically equivalent to ridge regression BLUP based on what kind of multivariate normal distribution you make about those marker effects. But I think this is a simpler way of looking at it. So in G-BLUP, basically you assume the individuals and their polygenic effects, their genetic background in other ways, in other words, that's considered here to be a random effect. And if it's considered to be a random effect, we assume that it has a certain distribution, a multivariate normal distribution that follows this covariance structure. And this covariance structure here is the G matrix. And basically in this G matrix, the elements here in this G matrix are basically the realized genomic relationships calculated using the markers. Okay? This is basically how everybody in your population relates to each other according to the genomic data that you collected using your markers. So you're basically sharing information between relatives in this mixed linear model, right? And we know individuals in different populations are related to each other more or less than with other individuals. We have individuals with different pedigrees. And we know that for polygenic traits, individuals that are more related to each other than other individuals are going to resemble each other more for complex traits, right? But sometimes there are deviations from this expectation. Sometimes individuals who have the same pedigree are quite different. You may have siblings, you may have several siblings. One of your siblings, you may look more like one of your sibling than another sibling. And that's because we have, in addition to relationships, we have what we call the Mendelian sampling term. The Mendelian sampling term. That's what causes individuals to basically be more or less related to each other, even if they have the same pedigree. If you look within a biparental family here, say this is a maize now, recombinant inbred line family. Individuals that are from the same family, of course, have quite differences in terms of their marker similarity. Some individuals only share 55% of the alleles. Some individuals share 85% of the alleles. All right? So we can model pedigree relationships, but the problem with only modeling pedigree relationships in a model like this is it doesn't capture this part of the variation. And markers allow you to do that. So the ideal matrix that you use in this model would be a relationship matrix calculated using all the causal polymorphisms. If we knew a trait, we knew every single causal polymorphism that controlled that trait, and we could track every single one, and we could actually weight those according to their effect on the trait, and then calculate this matrix. That would be the perfect matrix, right up there in the upper part of the slide. But we don't know that. We don't know hardly anything about complex traits in terms of the causal polymorphisms. So we estimate it using markers. And the degree to which we can estimate this very well depends on our linkage disequilibrium, of course, which depends on our marker density, which of course depends on the relationships or the diversity within the population we're working with. So this is an important concept I'll come back to in a little bit here, but I wanted to introduce it right here. Basically what this says is that of course, the expectation of the genomic relationship between two individuals is equal to the pedigree relationship. That's what you'd expect. You have deviations around that because of Mendelian sampling. And as that expectation goes down, the variance of those genomic relationships goes up across the genome. All right, so if you say that, well, let me go to the next slide here. So basically this is what this says. It says that if you have two individuals here that are distantly related, all right, their true relationships are going to vary more across the genome. They're going to vary more than individuals that are more closely related. All right. So I'm not going to go any further with that right now, but I'll kind of touch on the next topic I want to talk about. All right, and not this topic, the topic after this. All right, so now set that aside, let's talk about G-BLUP. Okay, genotype environment interaction effects. They're hard. Well, they're not that hard. They're actually pretty easy when you're trying to predict tested individuals or tested genotypes in tested environments. That's the pretty easy problem. It becomes less easy when you're talking about untested genotypes in untested environments, and when it gets really, really hard is when we want to predict untested genotypes in untested environments, which is really what we want to do as plant breeders. We don't really ever observe the environment that we want, the perfect target population of environments that we want to breed for. We only can breed based on what we've observed in the past, which may or may not resemble what we ultimately want in the future. So that's a hard problem. And when we have tested environments here, we can estimate G-BLUP effects. But when we don't have tested environments, we can't calculate a G-BLUP effect. The only way we can do this, basically, is to connect the environments through environmental covariables, weather variables, soil variables, that type of stuff, to tell us the similarity between our tested environments and our untested environments. How similar are they? All right, so this is a sort of a schematic of how some of these different options for predicting G-BLUP work. You have your training data, right, your genotypes and your phenotypes. You collect your phenotype. These are your observed environments. And you can feed these into a genomic prediction model and use this information two different ways. You can either feed it into like a crop growth model type of thing, and that will inform you how to weight the environmental variables in your genomic prediction model. It's basically kind of a biologically driven or physiology driven type of model to help you weight how important those environmental covariables are on something like yield, right? Or you can use what some people call this an environmental kinship model. All right, you basically collect all the environmental variables around your environments, weather variables, soil variables. You use all those to sort of tell you how similar environments are to one another, and that helps you make predictions in your mixed model there. All right, and then you predict your new target environments. So two different ways of looking at this. This is very biologically driven. This is statistically driven. This is statistical. This is a little bit more biological, although a little bit statistical. I'm only going to talk about the crop growth models one here for today. So this is a very nice paper that was published in PLOS One a few years ago by some folks at Pioneer, and they basically laid out this framework for using crop growth models to integrate into a genomic selection model for making predictions in future environments. Right, I'm not going to go over the details. The details are way beyond what I want to talk about here. But basically what they showed was that if they take this crop growth model and they use that to help predict G by E with all the genomic data and the environmental data, they get to do a lot better. This is outside the zone of who cares. This is doing pretty well, right? So 0.77 versus 0.54 for a crop growth model versus a G-BLUP model. All right, so their crop growth model in this simulated data set worked quite well, and it's a very nice idea, a very elegant model, all that kind of stuff. It's really nice. But you go to the next paper in their real data set that they collected on double haploid lines, and they didn't see much of a difference. So in this case, you compare the crop growth model to the G-BLUP model. You know, this is pretty minute differences here. Sometimes you actually do a little bit worse. So in a real data set, it didn't seem like they weren't getting quite the response that they had hoped. Either the signals that they expected in the crop growth model weren't the same, or there wasn't as much G by E, or I don't know what it was. They actually explain a little bit in this paper, but I don't recall all of it off the top of my head. But it didn't quite work as well as they expected it to. Okay, so that's it for G by E for now. Let's go into training population design. Quickly, how do we select an informative training population for genomic selection? So we all know, or the people working in this field know that your prediction accuracy here increases quite quickly as you increase your training population size. Just like your marker effect estimation accuracy, your your QTL detection power, that all increases with population size, right? This is well known. What we want to know is can we find small training populations that are more informative and result in a higher predictive model than training populations that are very, very large. This will cost a lot to phenotype. This would cost less to phenotype, and we could maybe select a subset here that's more informative. So a few points here. So the first point is that close relationships matter. Individuals closely related training populations to your target population is the best case scenario. Actually, your best, easiest case is if you work within a single biparental population. Take one part of your biparental population, phenotype it, build a genomic prediction model, predict the remaining part of the biparental population. That's the best, easiest case scenario. The problem with that as a plant breeder is that I don't want to wait to phenotype that biparental population before I make selections in it. I want to make predictions based on the rest of the data I collected in the past and predict new biparental populations. I don't want to wait around. So I don't like that. So it's useful to know that close relationships matter. Another thing here is that the genetic representation of your training population to your target population matters a lot. And this may seem like an obvious point, but it's still worth making because I get questions about this all the time. You know, your training population has to represent your target population. Here's an example from the literature. I don't have the citation up here. This is Windhausen et al. 2012. They basically took a diversity panel here of some tropical lines. They did a lot of phenotyping, genotyping, built a genomic prediction model, and applied this prediction model to a bunch of biparental breeding populations. And you can see that they did, actually going in the opposite direction here. They have negative prediction accuracy, so it completely failed. They put a lot of, I think part of the point of the paper was to illustrate this, but if you were to do this in real life, put a lot of effort into training a genomic selection model using a diversity panel to predict breeding populations, I think you're just going to set yourself up for failure just when you use it in this naive way. There's other things you could perhaps do to augment more relevant data types that we don't need to get into. This is from some of my own work, where basically we looked at genomic predictions across and Diego's actually in the audience here. He led this work across the germplasm collection in soybean, and you can see that within a cluster we get pretty good prediction accuracies, but when we try to predict across clusters, in other words across non-representative training populations, the prediction accuracy falls off pretty quickly. So that matters a lot. Thirdly, optimization algorithms can help select a training population. It seems like these things are pretty promising. So the idea behind these optimization algorithms is that you have a universe of available training individuals to select from. Maybe it's your entire germplasm collection, maybe it's a huge breeding population, all right? You want to basically, you have information about your target population, you know what you want to select on, so you have to feed that into this, and then you optimize the selection of this. And there are a few different ways to optimize a selection. You can either iteratively maximize your genetic diversity, and I think this one works really well here where you try to minimize the prediction error variance. Basically, the prediction error variance is something that comes out of the mixed model, and you can iteratively try to find a population that minimizes this prediction error variance. And the prediction error variance is going to be mostly dictated by your total population size and your relationships between your training population and your target population. So there's algorithms, there are genetic algorithms that can be used for this. There are basically these replacement algorithms that go through and march to a better solution and try to minimize these things. So you do that, you select your training population, you phenotype it, and hopefully your optimized training population has a higher prediction accuracy compared to a randomly selected training population. And it works quite well. This is just an example from some of my own work, actually in maize, but there's lots and lots of publications out there that show that you do get a pretty good response when using these optimization algorithms for selecting a training population. So I would recommend looking into those if you're interested in this, applying this. So that is basically what I have for kind of the overview. How much time do I have here? 2 minutes, okay. So just a little bit about this project we have going. Like I said, there are only a handful of publications so far in soybean. I think this project here, funded by the North Central Soybean Research Program here, is going to help change that situation. And the title of the project is to increase the rate of genetic gain for yield in soybean breeding programs. And there are several objectives on this project. Objective 2, which is the one I'm mostly involved with, is basically the gist is to enable and study genomic selection for soybean. And there's lots of people from the North Central region on this, a lot of Iowa State folks like Sylvia and Bill Beavis, Danny Singh, yes. So the idea is we have a lot of historical data. We want to combine that with our genomic data and enable prediction. And one place we're looking for historical data is in the uniform soybean regional trials. These things have been conducted for quite a long time. It's basically a high quality, deep set of phenotypic data that we can use. A lot of breeding lines evaluated at 5, sometimes as many as 12 locations every single year. So it's a good set of data. And we're working on genotyping this data, basically going back to breeders throw away their seed, unfortunately. So we can't get all the seed of all these lines going back, but we've had some success going back to 2006, getting seed. And of course, as you approach current day, then our chances of getting seed are better for genotyping. And we've spent a lot of time cleaning and both phenotype and pedigree data from the URTs going back to 1989. And that's about ready to be analyzed, uploaded up into a database. And the whole gist of it here is to help leverage this historical data and maybe leverage some resequencing data with some 6K data and come up with a scheme for merging different types of, I didn't talk about the markers, but you can merge lots of different types of genotype data sets to try to unify things a little bit, ultimately with helping with predictions. So develop a training database, develop methods for unifying genotype data from various platforms. David Heighton is working on an ultra cheap and ultra easy genotyping method, and we'll eventually test this out in individual breeding programs a couple years down the road from here. And I should mention that Matt Hudson is working on this sub-objective in the project. So in summary, lots of advancements in genomics applied to breeding and genotyping. I think a lot of advancements since the last World Soybean Research Conference. Lots of advancements in the development and adoption of statistical models. But simple models like G-BLUP still seem to rule the day. I think that predicting genotype and environment interaction effects has a lot of potential that hasn't been realized yet. And I think that's a huge area, a huge frontier that we should spend more time on as a community. And we need to continue to refine training populations. And I think an important thing with the training population piece is that first, you need to define the target population and then define your training population. I think that's the take-home message. And we need to continue to build these data sets so we can continue to work on these problems together. That's it. I just wanted to highlight the people who worked on the data in this presentation would include Ben Campbell, who leveraged this project to get an NSF fellowship. Dineshwar and Diego, who's in the audience, and Jen Floor, my lab technician.
So you mentioned there's 8 studies out there in soybean that have used genomic selection. Are these? Can you give us a little overview of what traits? Is this simulations, or what kind of studies are out there? What have we learned so far about what it can do for soybean?
I mean, most of those are on white mold resistance and yield and plant height, soybean seed, stuff like that. Basic. Both yield and also what we would consider to be kind of model quantitative traits. And the predictive accuracies that we see are, it depends on the study, but they range anywhere from, well, I think it was somewhere from about 0.4 to about 0.7, wherever that is, right there. So, some pretty high prediction accuracies, some pretty low ones. I'm not sure which ones are yield and which ones are plant height in here. But I would say that we're still sort of figuring that out. I would say that we've learned that we can calibrate and train fairly predictive models. I think there's a long ways to go yet, and I would say that none of these studies in soybean have looked at many of the optimization questions that I've talked about. I would say it's more of sort of just let's see what it looks like and then go from there.